So today we will start with the bifunctional enzyme. Name itself indicate two functions or two chemical reactions happen. So bifunctional enzyme that possesses are two distinct catalytic activities. Two distinct catalytic activities at active site or multiple active sites where you will find a single polypeptide chain. These enzymes, what is the functions of these bifunctional enzyme? So these enzymes can perform two different chemical reaction as I say by using the same active site or different active site. So the presence of multiple reactions can be happen with a single enzyme, and it shows the efficient channeling of substrate between the consequently reactions in a metabolic pathways. So, in short, we can say it has a crucial role in many biological processes. Now, what are the example? You can find in the biological, our body also. DNA repairs. How the DNA is repairing through so same bifunctional enzymes. I will give you one of the example of more number of example after this. Amino acid metabolism, there are certain enzymes that having a bifunctional enzyme. Then lipid biosynthesis is also included bifunctional enzymes. Now another terms that is pseudoenzymes. Pseudoenzymes means that is not actual enzymes, but it can be created with the mutations. It has no functions, but sometimes you can use with genetic engineering and making for the particular purpose. So pseudoenzyme itself also a protein, and that is resemble or it is near to the enzyme, but very important characteristic, it lacks catalytic activity. Despite this enzymatic activity or enzymatic function, it has an important role in regulating cellular signaling pathways. Pseudoenzymes can act as a scaffold, or it can also used as a inhibitors to modulate some activities as a counterpart. These protein or pseudoenzymes evolved from an ancestral enzymes through genetic mutation, as I said previously, that render the catalytic activities. Now I am giving you switch over to bifunctional enzymes and their example. So alcohol acyl transferase AACT, this enzyme function as a bifunctional that catalyzes both. There are two reactions can catalyze by AACT. First is esterification, another is alcohol lysis in the fatty acids. So it will producing acyl CoA and fatty alcohols. Similarly, you have also found in the glycolysis. You know that reaction, aldolysis. It has also a bifunctional enzyme and it catalyzes both aldol reaction as well as dehydrogenation reaction of any sugar, glucose 2. So it will convert producing into a glyceraldehyde 3-phosphate and dihydroxyacetone, which is the very important functions you will find in the glycolysis. Then amidohydrolase, that is also found in the plant bifunctional that catalyze both hydrolysis and amidation. Amidation means of amino acid producing alpha amino acid and alpha hydroxy acids. Similarly, fourth example of bifunctional enzyme is acyl CoA synthetase. This catalyzes the activations both activation of fatty acid to acyl CoA, and you can also found deacylations of the acyl CoA to fatty acids. Another example you will also found in the citric acid cycle, or you can say Krebs cycle, malate dehydrogenase. It has also bifunctional enzyme and that catalyze both oxidation to malate to oxaloacetate and reduction of oxaloacetate to malate. Same enzyme can work in the both two products. Similarly, we will continue with that. Another example you will find in the plant is nitrogenase activities that catalyze the reduction of nitrogen gas N2 to ammonia NH3 plus, as well as it will give you a oxidation of ammonia to nitrate. Remember these bifunctional enzyme activities. Then PEPCK, phosphoenol pyruvate carboxykinase. It has also catalyzed bifunctional enzyme activities that PEP, conversion of PEP to oxaloacetate, as well as to conversion of carbon dioxide to oxaloacetate. Same bifunctional enzyme pyruvate kinase, quinone reductase. For pyruvate kinase, you will find pyruvate to acetyl CoA and conversion of acetyl CoA to pyruvate. Then quinone reductase that catalyze both reduction of quinone to hydroquinone and the oxidation of hydroquinone to quinones. Sucrose synthase, that is bifunctional enzyme that catalyze both conversion of sucrose to glucose and fructose and conversion of glucose and fructose to sucrose. So these are the examples of bifunctional enzymes. And it has many important roles, as I said, in as per the glycolysis. It means in carbohydrate metabolism, amino acid metabolism, lipid metabolism. I will not put up to a plant. I will give one more example of that. But before that, we will understand what is pseudoenzymes, how it's work. Pseudo name itself, it is not an enzyme, but it is a protein. But it can have a role. That is also, some people said that are not found in the nature. But I can prove that it is found in the nature also. So I will give you in the both condition in animal and plant. Now we will start with the plant, how it can utilize for the human mankinds or in agriculture productions. So pseudoenzymes are enzymes that are not found in nature but are created artificially through genetic engineering, or you can use another method too. Pseudoenzymes play an important role in the major functions in improving a crop yield, increasing a resistance to disease and pest. It will also enhance certain nutritional content or nutritional quality. So I am giving you here certain pseudoenzymes which are used in the plant crop improvement. One of the greatest example is herbicide tolerance plant. You can produce with the genetic engineering. Nowadays the market have certain plant too. So these enzymes are designated to confirm a tolerance to herbicide, and that allowing you can put or farmers to use these chemical to control weeds without farming their crops. For example, the herbicide tolerance pseudoenzyme. What is the name of that? That LPX401 was developed by this company, and it has been used in the soybean, corn to increase our crop yield by reducing the herbicide. Okay. Now another example is insect tolerance pseudoenzymes. You can say chemical. Okay. Disease resistant pseudoenzyme. You can say disease resistant chemicals. Nutritionally enhanced pseudoenzymes. So pseudoenzyme can use to make a crop more resistant to insect pests. You have pseudo carbamazepine. Okay, Bt toxins. That was developed by Monsanto, and that uh, our example in Gujarat, they are reached to a greatest production of Bt cottons to protect against bollworm. Yes, another pseudoenzyme RPP1. Okay, that is disease resistance, and it is also a vector that is used in the rice. Similarly, another golden rice, golden egg, improved golden. All you can say that the pseudoenzyme is a gene which is inserted for improvement of certain crop. Okay, golden rice. Hmm. So improve the dot tolerance, increase crop yield. One of the example. Now I am giving you a key clue point of biochemistry and molecular biology or biotechnology. Real pseudoenzyme examples. That was the end product. Okay. So list of pseudoenzymes. Enzymes are a protein that I have repeated many times that resembles but lack a catalytic activity due to mutations in their active sites. Why pseudoenzyme does not have activity? Because it lack a catalytic activity, because it can create own self at mutation at active site. Therefore, it doesn't give it. These pseudoenzymes play a crucial role in regulating various cellular processes. So here are, as I see the example, you have to remember these enzyme activity. Both are true. For example, P38 MAP kinase, that is full form of mitogen activated protein kinase. Mitogen activated protein kinase is a serine or threonine. These are the two amino acids. Kinase is the enzyme. Serine or threonine kinase that regulate the cellular response to stress and inflammation in the living beings, or you can say human beings or animals. However, variant of these P38 gamma lacks kinase activity, make it pseudoenzyme. Despite these, that interact with the protein and influence signaling pathways. Now, another name itself pseudoenzyme, pseudokinase. So, pseudokinase is a diverse group of proteins that, if you see, that structurally resemble to kinases, ATP kinase. You know very well. But here we are dealing with the pseudokinase. Lack the ability to phosphorylate substrate. What are the example of pseudokinase? Including JAKs. This JAK's name is Janus kinases, which contain a pseudokinase domain that modulate the activity of kinase domain. Now, another, not up to this, we will include a pseudo phosphatase, pseudo proteases, pseudo glycoside transferases, and pseudo enzyme itself. You know very well. At concluding remarks, I have given three lines. So pseudophosphatase also mimic the structure of phosphatases, but do not possess the phosphatase activity. Same is pseudoproteolytic activities in pseudoproteases, which contain a mutated active sites and function as a regulator of the proteins. Then pseudo glycoside transferases, glycoside transfer, but do not possess the glycosylation activities. For writing in the examination, you need not to remember. You have to remember only a name of enzyme, and you have to write like that. Do not possess or lack of activities. at active site mutations. Same thing you have to remember for pseudo enzymes and bifunctional enzymes that already been taught to you. So this is all about the